Cellular respiration, we're going to start by looking at glycolysis. So overview of cellular respiration, and we know that the general formula is that you take glucose, you add some oxygen to it, and it gets converted into carbon dioxide and water, and the point being that we get some energy out of it. So simple equation to write down the overall process, but we're going to be looking at the details of this equation here. First one that we are looking at in this video lesson is glycolysis itself. And so this is the very first part. This is taking glucose and converting it into pyruvate. And we will get a little bit of this ATP energy out of it. Second stage, we'll take that pyruvate, it'll oxidize it and get us to this sort of intermediate acetyl-CoA molecule. We then run that molecule through this cycle called the citric acid cycle or Krebs cycle, and that will produce some more ATP as well as a bunch of electron carriers, which are useful when we go to do the last step, which is the electron transport chain, where we are converting all the electron carriers we've created into more ATP. And remember, each time we are producing this ATP, that is the energy that is the reason why we're doing this entire process to begin with. Remember that prokaryotes don't have any fancy organelles. They have their cytoplasm here, and they evolved in an anaerobic environment, atmosphere. There was very little oxygen on Earth when prokaryotes were evolving, and so the process that they use to get energy, one of the processes that some of them use to get energy, does not require O2. That's glycolysis. And since us and other eukaryotic organisms like us have come from prokaryotic cells that have endosymbiotic theory and all that evolved into these eukaryotic cells, we can still see this glycolysis remnant that was adapted from this anaerobic environment that prokaryotes developed their ability to get energy from. We can still see this as part of our cellular respiration. Yes, we have access to oxygen now, but our energetic processes still involve this anaerobic part to the process. And this is it. This is half of it. Anyways, this is glycolysis 1. So we break it down into two parts, 10 steps broken into two parts. Glycolysis 1, this is an overview, and then we'll look at the details of it. But we're starting off with a glucose molecule, and we are splitting it apart, hence glycolysis. We're splitting the glucose apart. And if you look at this, we've got our first step, our second step, our third step, and our fourth step right here. This makes up glycolysis 1. And if you think about it, if you look at what's happening here, and we'll look at this in more detail, we are using up ATPs. We are using up energy. We're breaking them into ADPs in glycolysis 1. So this is why we separate from glycolysis 2. Glycolysis 1 actually requires energy. The end of glycolysis 1 ends up with these two molecules called G3P or glyceraldehyde 3-phosphate. There is a little step in here that we'll look at the details of. It creates one type of molecule and then forms G3P. So here is the process in detail. As you know, this is, we've got down, down, up, down for our OH groups on our glucose monomer here. So here we have our alpha glucose. And the first thing that happens to it during glycolysis 1, or the first step here, is that ATP is reacted with it. And again, this is done through enzymes. So the enzymes are listed here for each step. So hexokinase will take glucose and ATP and turn it into glucose 6-phosphate, which isn't such a bad name to remember because glucose gets a phosphate on the, remember this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and it's carbon 6. So since we are putting a phosphate from the ATP onto glucose 6, this would now be called glucose 6-phosphate. And there it is, phosphate on the 6 carbon of glucose. It is now called glucose 6-phosphate. It then goes through the next step. And again, another enzyme will take glucose 6-phosphate. And it is making this isomer of glucose 6-phosphate called fructose 6-phosphate. So you can see that we've sort of essentially unattached the oxygen here from this carbon. And so we are down now to a pentagon shape with the oxygen in there. Nothing has been added or taken away. It is just turning into a different isomer. So this is not glucose 6-phosphate, but instead fructose 6-phosphate. Once we have fructose 6-phosphate, we can then add another ATP. So this is the second ATP that is being destroyed. Not a very good idea when you're trying to make energy to be using all this energy, but this is the beginning part. And so we take this ATP and we are going to then also add on a phosphate onto carbon, wrong one, carbon 1, 2, 3, 4, 5, and 6. And so we now have a phosphate on carbon number 1, and we still have our phosphate on carbon number 6. So, nice name for this one. It used to be fructose 6-phosphate, is now fructose 1,6, because there's a phosphate on the carbon number 1 and the 6. So fructose 1,6, and there's two of them, so we say bi, or actually bis, phosphate to say that there are two of them. Once we have produced fructose 1,6 bisphosphate, another enzyme is going to split it apart, and we're going to get two products from this point going forward. Just a quick side note here. Phosphofructokinase is an important enzyme for the regulation of this process. So if you did want to shut this process down or slow it down, this is a good enzyme to target. The two products that we get from our fructose 1,6 bisphosphate are glyceraldehyde 3-phosphate, that's the important one. And then we get this other one, dihydroxyacetone phosphate, which then gets converted into another G3P, glyceraldehyde 3-phosphate. So essentially we have two of these glyceraldehyde 3-phosphates, which are, again, we've got these 3 carbons. One of them has got this phosphate group on it. One of them has a hydroxyl group on it, and the other one has a double bonded O. And then you fill in the rest of the bonds with hydrogens, which we don't care that much about in biology, but it would make it stable. And so that is our G3P. G3P, glyceraldehyde 3-phosphate. You can kind of remember it looks kind of like a glycerol molecule, but it has a phosphate on there. We've got this aldehyde group on there as well, so not too bad to remember that name glyceraldehyde 3-phosphate. So the third carbon has a phosphate group, and we've got an aldehyde on the end. So now we have two of these, because remember, this dihydroxyacetone phosphate is no longer, it is now a second. So now we have two G3Ps, we got one there, and we've got one there. So everything from this point on in glycolysis is going to be happening to two molecules in tandem. Glycolysis 1 overview of the whole process itself. We have consumed free energy. We've used up some ATPs. Not good in the overall process. This is like taking 2 steps back. But summarizing it, we took our 1-glucose, which again has one of them with 6 carbons in it, and we've destroyed 2 ATPs that became ADPs. They've lost their phosphate groups. Each one has lost its phosphate group. And we now have 2 G3Ps, glyceraldehyde 3-phosphates, which are going to enter the second part of glycolysis, glycolysis 2. Just a quick note, whether or not you have oxygen present or not, glycolysis is all going to be the same. Glycolysis does not require oxygen. The second half of glycolysis, called appropriately glycolysis 2, again, you need to think of it as, often the diagrams will have one of these chains represented, but realize there's 2 of them occurring in tandem. We're starting off with our glyceraldehyde 3-phosphate, of which we have produced 2, making sense because we had 6 carbon glucose to start, and now we have 2 3-carbon glyceraldehyde 3-phosphates. This is the overview. As you can see here, we're going to be producing some NADHs, which, if you remember, those are the electron carriers. And also, very importantly, we're producing ATPs, because so far we're in the hole by 2. But as you can see, by the end of the process, we're going to be up. We're going to make 4 of them, leading us with a net gain of 2 ATPs, plus these 2 electron carriers. All right, here is the one view. Do realize, though, there are 2 of these happening at the same time. So there's 2 of these series of reactions happening in tandem. Glyceraldehyde 3-phosphate is going to, again, through the help of an enzyme, it is going to be changed into 1,3-bisphosphoglycerate. And so we still have the 2 phosphate groups on here, but what's happened is it has lost some electrons, and this NADH now has, so it has basically picked up electrons from our G3P, turning it into 1,3-bisphosphoglycerate. Phosphate is being added to the first carbon on here. You notice that each G3P only has 1 phosphate. This is now, remember this carbon number 1, this, let's put that carbon number 1, carbon number 2, carbon number 3. So it was G3P before, the phosphate was on the 3rd carbon, and now we still have that one on the 3rd carbon, plus we have this extra phosphate group on the 1st carbon. So the name 1,3-bis, referring to 2 phosphates on there, so bisphosphoglycerate. This is then going to be used to make an ATP. So this is where we're actually getting some ATP out of it, getting some energy out of it. Another enzyme's involved in that process, and it's going to remove a phosphate. So you notice that we are down to just this top phosphate here on our 1, 2, 3, 3rd carbon. So instead of being 1,3-bisphosphoglycerate, we have lost the one that was on carbon number 1, so its name is now appropriately 3-phosphoglycerate. We've lost the 1 and the bis. We don't have 2 of them. And so now we just call it 3-phosphoglycerate. And that's because this ATP that we just made now has the phosphate attached to it. Then, next step, another isomer. It is going to be just rearranging this molecule. And so it goes from 3-phosphoglycerate. It was on the 3rd carbon here. Now it is on the 2nd carbon. So appropriately, its name has gone from 3-phosphoglycerate to 2-phosphoglycerate. That's, again, just an isomer of it. So this mutase name in the name helps you recognize that as an enzyme, because it ends in A's. But mutase means it's isomerizing it. It's making another isomer of it. Then we're going to take our 2-phosphoglycerate. We're actually removing a water from it with another enzyme. And the water comes out, we end up with this molecule here, phosphophenol pyruvate. Almost at the end here. It still has the phosphate on carbon number 2. It gets removed to make our another ATP. And remember, each of these are times 2. So we have now produced 4 ATPs. And once that phosphate is removed, we are not at phosphophenol pyruvate, we are just at pyruvate. And so this 3-carbon molecule here, we have just hydrogens at the top, we have a double bonded O in the middle, and then we have this hydroxyl group, which if you take the hydrogen off, we just have an oxygen and a negative down at the end there. Sorry, I should have said carboxyl group at the end here. And this is our pyruvate molecule, which is the end product of glycolysis. Remember, there's 2 of those because we've run this second half, glycolysis 2, twice. So we have produced 2 pyruvates and 4 ATPs, plus those 2 electron carriers. So overall summary for it, we have 2 glyceraldehyde 3-phosphates going into glycolysis 2. They have now turned into 2 pyruvates. We've loaded up 2 electron carriers, so we've created these 2 NADH versions, which are the version that's actually carrying the electrons. We've made 4 ATPs, but remember we used 2 of them when we did glycolysis 1. And we also got 2 waters out of the process. We have actually produced free energy in the second part of glycolysis. Overall, a net gain of 2 ATPs because we had to pay 2 in to get these 4 out.